Nuclear Energy, Key to Tomorrow. Produced by WQED for the Educational Television and Radio Center. In cooperation with the Westinghouse Electric Corporation and the Atomic Energy Commission. The subject, agriculture. Your host, Dr. Warren F. Witzig, Manager of Reactor Physics, Bettis Atomic Power Division.
Welcome to another discussion on nuclear energy, key to tomorrow. In some of our past get-togethers, we've talked about the application of nuclear energy to power production. We've talked about the application of radiation in industry and in medicine. In this discussion, we're going to talk about the application of radiation in the field of agriculture. Naturally, we are concerned with maintaining the high standard of living that we have in this country today, and we are equally concerned with ensuring that we maintain this high standard of living in the future. To accomplish this, we are going to have to do a lot of research and development work in the field of agriculture. Some of this work will include learning how to make plants develop and grow in regions of low rainfall, in regions where we have unusual temperature conditions. And further, we will have to develop plants which are disease resistant. For this discussion, we have with us a geneticist, Dr. Richard S. Caldicott, who is with the Cereals Crop Branch of the United States Department of Agriculture. Dr. Caldicott is stationed at the University of Minnesota in a cooperative agreement. Dr. Caldicott is going to talk with us about this application of radiation in the field of agriculture. Supposing we go into Dr. Caldicott's laboratory and visit him, see how he proposes to do this. Dick, are you ready for us?
Yes, Warren, I believe I am.
Fine.
I think if we're going to discuss this subject intelligently today, that perhaps we had better review some elementary but quite important biological concepts. Let us take a look at this process of cell division, because it is the subject which will concern us most and will serve to introduce our topic best. As most of you are probably aware, the cells of all biological systems, with the exception of a very few bacteria, are very much alike. They consist, for the most part, of a nucleus and a cytoplasm. The nucleus is the part of the cell which is of greatest concern to us today, because it is the part of the cell which contains the genetic material which makes organisms develop into whatever they develop into, makes them respond to their environment in the manner in which they respond. I would like first to introduce you to what we call a non-dividing or a resting cell. In the resting cell, the nucleus appears to consist of long reticular strands wound throughout the entire volume. When this long reticular strand starts to shorten and to thicken, which it does by coiling, the cell signals to us that it is undergoing the process of cell division. And this coiling and this thickening progresses to the point that the long reticular mass ends up in a few very distinct, easily detectable chromosomes, easily detectable in that we can see them with the light microscope. And when the cell reaches this particular stage, we say that it is in the prophase of cell division. At some point between interphase and prophase, the chromosomes have doubled throughout their entire length, some point during this coiling and thickening. When these well-defined chromosomes progress further towards division, when the cell progresses further towards division, these well-defined chromosomes line up on the equatorial plate of the cell. They line up along the middle of the nucleus, which we know as the equatorial plate. When they line up at this point, the two poles in the nucleus, the two extreme tips, which I'm pointing out here, will send out a spindle fiber, as we call it, a fibrous region which attaches itself to the two-stranded chromosomes and pulls one half to each pole. And as the poleward movement progresses, we call this particular stage the anaphase, the anaphase movement of the chromosomes towards the poles. When anaphase movement is complete, a constriction or cell wall will develop through the middle of the entire cell, including the nucleus, and end up with two cells that look very much like the original interphase cell we discussed. The chromosomes that had such sharp definition lose their clarity. They go back into the very similar reticular state that they were in the original interphase cell. Now, I would like to point out to you that the individual chromosomes carry along their length discrete entities which we know as genes. And it is the action of these genes that gives to an organism all of the characteristics that it expresses. It determines whether the organism may produce red flowers or white flowers, whether the plant may be tall or short, and the like. Furthermore, these chromosomes come in pairs in all of the body tissue that we look at, with all of the plant or animal body tissue that we see with the naked eye. And these pairs of chromosomes have originated originally from one chromosome coming in from one parent and one from a second parent. Now, we will later be discussing the chromosomes of the barley plant. And in this discussion, I will point out to you that 7 chromosomes have been originated from the female parent and 7 from the male parent. And each one of these 7 chromosomes that comes from one parent has an identity, an exact equal in the 7 chromosomes that come from the other parent. And these chromosomes, as we will discuss later, pair up during the sexual stages, or the stages which just immediately precede the development of eggs and sperm. And this will be of some significance to us, to remember that the chromosomes have an identity, one coming from each parent.
Dick, let me just make sure that our viewer understands this. Now, we're going to take a look at how the radiation affects these chromosomes and the genes.
That's correct.
Fine, fine.
Let us go over here where we can look at some of our illustrations, just to give the group an idea of the kinds of things that radiation does to a biological system. Now, I have here a number of dishes in which barley seedlings are growing. Over on the extreme left are seedlings which have not been subjected to irradiation. These we would call the control seedlings, and the height of these seedlings represents normal development for these particular plants. In these other groups, we took dormant seeds and subjected them to different doses of irradiation. This group here was placed for 1 hour in the thermal column of the Brookhaven National Laboratory's nuclear reactor. These were in for 2, these for 3, these for 4 hours. After irradiation, the seeds were placed in these dishes and allowed to grow in this way. And you can see that as the dose of radiation increased, the amount of injury that the seeds incurred also increased. Now, I would point out to you that this injury that takes place and that is a result, that is the reason for these seedlings not growing as well as the control, is entirely due to damage which has taken place in the nuclei and within the chromosomes of the seeds that were irradiated. In other words, the cell division rate has been retarded if cell division has not actually been stopped by the irradiation.
So this is an example now of the effects of radiation on chromosomes themselves. not the genes.
It's for the most part on the chromosomes, but it would also, genetic changes, gene mutations would have a part in this reduction.
So it really is an effect on both of these.
Yes, I think we could say that it is an effect on both of them. Let us look at what the chromosomes in a barley plant look like and the kinds of changes which ionizing radiations can bring about in these chromosomes. Now, a normal chromosome in a barley plant at the metaphase of cell division, one of those stages we talked about a few minutes ago where the chromosomes line up along the middle of the cell just before they prepare to move to opposite poles. These chromosomes normally look like these little V-shaped configurations I've shown you here. They have one prominent region in here which we know as the centromere. It's this region to which the spindle fibers attach and which guides the chromosomes to the poles. In other words, it causes the chromosomes to separate. Now, if one subjects these seeds to irradiation and breaks off a piece of chromosome, as for example a piece of this chromosome, and one also broke off a piece of this chromosome, it's possible for the two parts which have centromeres to join up. When they join up, one can obtain one long chromosome with two of these regions in it. This chromosome here no longer behaves normally during cell division because instead of having the one centromere, it has two. And this, to this particular chromosome, is an exceedingly confusing event.
So it could be pulled to really both poles, then,
could be pulled to both poles at the same time.
Uh-huh.
Now, I can illustrate this for you in our next figure. Here we have the anaphase, an anaphase of cell division, again in a barley cell. The pole on this particular cell will be one pole here and one pole here. And the chromosome that has two centromeres on it is this particular two-stranded entity here that is being pulled towards the two poles. That's the type of thing that happens when chromosome structural changes take place that involve more than one centromere. So that the ultimate fate of this cell would undoubtedly be death. Now, I mentioned a few minutes ago how the chromosomes that come from each parent, which are identical from each parent, pair up at certain stages in the life history of an organism, just preparatory to the production of the egg and the sperm. That is, one exact chromosome that came from one parent pairs up with the exact image that came from another parent. The reason they do this, to the best of our knowledge, is because they are intimately alike throughout their entire length. And if one changed the structure of these things, they would no longer be able to pair up. And I want to illustrate this to you. For example, if we were to have broken with radiation a piece of chromosome off here and attached it down here, this chromosome would no longer be identical to this chromosome. So it would no longer pair in the fashion that I've evidenced here. It is possible to actually induce enough changes to get these chromosomes rather badly broken and exchanged. And we can show this, Warren, in the next figure to give the audience a picture of what we're talking about.
And now these are the normal ones, just like we saw in the previous.
These are the two, one normal here and one normal one here.
Half of which has come from each parent.
Right.
Mm-hmm.
Now the other chromosomes which should have formed these normal little circles.
And that's in the case of barley now. We should have 7 of those.
We should have 7 total of these little circles. We only have 2. So 5 of the circles
are missing
and have formed up into this very large ring.
It looks something like a pretzel.
And this very large ring has come about as a result of these exchanges between the chromosomes because they're no longer exact identities and they can no longer pair with one another.
Now this is a case where I've taken radiation to produce a damage to the chromosomes such that the 5 which would normally be here looking like this have combined into this, well,
Very
this one
long-
very irregular type of thing. Now, this is the chromosome type of damage.
This is the chromosome type of damage, yes.
Uh-huh.
Now, I should say that this type of change is a very common kind of change. It's the type of genetic change which causes sterility in most of the organisms which become sterile as a result of irradiation. It's because the chromosomes can no longer behave in the same fashion.
Mhm.
Now, we're going to mention this afternoon something about mutations, the kinds of changes which occur within very, very small regions in the chromosome, and which we are unable to detect except through growing out the plant and having a look at it. In other words, they're very subtle changes.
To see what the children of this one do,
Right.
and their children, and so on.
That's exactly right. They're very slight changes which have taken place, reorganizations in the genetic material of these chromosomes. And I'd like to show you a figure that will give you an idea of the types of changes that have been induced in wheat by some Swedish workers. All of them are attributable strictly to a small change within the gene on the chromosomes.
So now this is the example of the changes produced on the genes by radiation, in contrast to the changes produced in the chromosome only.
That's quite correct.
Mhm.
Now, the normal wheat plant that they started out with was this one right here. They irradiated seeds of this plant, then grew the seeds to mature plants, took the harvest from those seeds, and from the progeny, from the original irradiated seed, they were able to get all of these different types of plant growth. For example, they were able to get a gene change that increased the height of the plant. They were able to get a gene change which gave them a roughly comparable height, but gave a rather different type of head structure. It's more compact here as opposed to here, and so on through these other different height variations. Now, I would point out to you that harvesting procedures that farmers undergo are often eased by having certain characteristics of head types on plants, by having certain heights of plants, so that some of these things can have some agronomic importance, some agricultural importance for these reasons. For example, Warren, this very tall plant here would be of little use if one had a high rainfall area with a high nitrogen in the soil, because the plant would grow so tall that it would tend to lodge over.
Mhm.
And the farmer doesn't want this when he's harvesting his grain.
Now,
So that,
that'd be a relatively useless crop, though.
yes, but on the other hand, under very dry conditions, he wants a plant that'll reach some height so that he can work with it with his equipment, so it might be valuable under those conditions.
Mhm.
The same general situation obtains for all of these
Yes,
other...
just the reverse situation, that if you had a very wet and a very rich soil, you might want one of these that grew considerably shorter, so that it would be just about the right height under these very favorable conditions for the thrasher. And likewise in the pods, that is the mechanical manipulation of the thrashing process could be aided by, for example, the loose pod versus the rather tight.
Yes,
pod.
this...
So here we can see a rather immediate application, can't we?
Yes, we can. And I think that I should emphasize here that all of the changes that we are talking about at this particular time are hit or miss changes. We're unable to predict with any consistency how often we'll get these from our radiated material. We have no way yet of directing mutation, of pinpointing mutation.
Well, this illustrates the very urgent need for a great deal of research in this field.
This, of course, is the point which I am personally most interested, not in whether or not we can get a specific desirable agronomic characteristic out of the work we're doing, but what are the kinds of things which control rates of mutations, types of mutations.
Being able to understand why.
Being
in
able
the
to
broad
understand
sense.
why. If we understand why, we can very soon put it to work for us. Now, I would like to spend a few moments giving you some idea of some of the biophysical studies that we have been conducting. Some of the studies where we're controlling the amount of oxygen that we put into our biological system by the amount of water we use in the system, and things of that nature.
Now, these again are attempts to understand why. That is the research of the field, right?
Yes, that's correct. Try and understand the whys and the wherefores of all of the genetic changes that we've discussed. I can tell you about one very interesting piece of research that is currently in progress. If we look again at this dish on the left, this is the control unirradiated seeds, seedlings that have grown from unirradiated controls, as we call them. All of the other seedlings here, received the same dose of X-rays, but were treated after irradiation in a little different way. This group here
sort of a way of developing a radiation protection, in a sense.
At least a radiation modification.
Modification.
I'm a little bit scared of the word protection in this regard. These seeds here were given the same dose of radiation, as I said, as all the others. They were taken from the X-ray machine and put into water into which oxygen was bubbled. So they were actually soaked in water that was enriched in oxygen. These were soaked in water that was enriched in nitrogen. Now, the time they were soaked in this water, before being placed in dishes to grow as you see them here, was 30 minutes in the case of these two.
Yes.
In the case of these two, the time they were soaked was for 4 hours. 4 hours in the presence of oxygen, 4 hours in the presence of nitrogen. Now, there are two significant points to this particular photograph here. One is that in 30 minutes, in the presence of oxygen, we had done as much damage as we could do in 4 hours.
Yes.
All of our studies have indicated that around 30 minutes gives us the maximum amount of injury. The point that interested us so very much was that the physicists and the biophysicists had indicated to us that the radiation damage was over and done with as soon as the X-ray machine was turned off. We now know that this is not the case. There are some after effects of radiation.
And you can influence
and we can influence them. This is a very exciting thing to us.
Has many implications.
It has some very profound implications. Now, our work that we are pushing very hard at the present time, is to get an indication of the kinds and frequencies of chromosome changes, the chromosome breaks that you have seen,
Yes.
and the mutations that occur in these different classes of seedlings.
Yes.
Because if we could show that we get different kinds and frequencies in one class than the other class, we may have some clue as to what the mechanism of bringing about these changes is.
Now, the nitrogen case, this is the 30...
30 minutes
and
soaking
that's
the presence of
the,
nitrogen. This is the 4 hours.
And I noticed that as far as height goes, it compares very favorably with the...
Very favorably with the control.
With the control.
These, I should say, were given a relatively low dose of radiation. We could modify these by increasing the dose of radiation.
Yes.
For example, had we given them twice as much, these would have been about half that height, and these about half their height.
But then we see, though, that for this dosage that these seeds receive, that there is an appreciable recovery from this effect.
At least the effect wasn't allowed to be enhanced by the oxygen, I think. And I should mention, I would like to show you another figure concerning this work, and I should mention that these seedlings that I've shown you had only 4% water in the embryo. And this is a very critical point. They were seeds that had been stored over phosphorus pentoxide, which is a very strong desiccating agent, before they were soaked in water. We were very interested in what would happen if we had seeds with a higher water content and treated them in the same way. That is, soaked them in the presence of oxygen or in the presence of nitrogen. And this figure here will give you some indication of what we're talking about. On this axis here, I have plotted seedling height in percent of the control. The unirradiated seeds would be 100. And as they received an increasing dose of radiation, the height of those little dishes of seeds that you saw a few minutes ago would decrease. This is the increasing dose of radiation on this axis, 5,000 R through 45,000 R. Now, if we take seeds with 16% water in the embryo, and we soak them in the presence of oxygen or nitrogen, we see that there is no effect of the oxygen treatment or the nitrogen treatment on the growth of these seedlings.
Now, this silking has taken place after the seed has been...
a post-radiation or a following irradiation in all cases, yes. So we see that with that wet seed, oxygen and nitrogen has no effect. And with the dry seed, as I indicated to you on that figure, when the seeds have 4% in the water in the embryo and they're soaked in the presence of nitrogen, they're less injured than when they're soaked in the presence of oxygen. Now, there are 2 points in this that interest us very much. One is that this curve here of the very dry seeds soaked in the presence of nitrogen always overlaps this curve here for the very wet seeds soaked in the presence of nitrogen or oxygen, as it does not matter with this particular class. It always crosses over here at a dose of around 35 to 45,000 R. We have no explanation for this. We're very interested in it. There's one other point that interests us a great deal. We know that the desoxyribonucleic acid, this is the protein-like substance of which chromosomes are made. We know that it changes its stereochemistry when it goes from 4% to 16% water content. So we're most interested in what is the effect of the oxygen on this chemistry when the seeds have a very low water content. How does this relate to when the seeds have a very high water content? Why is one sensitive and one not sensitive?
All right, Dick, let me see if I can recap this now to make sure that we have an understanding of it. We've seen the illustration of damage by radiation on chromosomes. We've seen it on the effects on genes. For example, those patterns of the wheat height and the size of the pod was a direct gene damage type of thing. And we see here on this chart that you just talked about that there are ways that we can modify this damage depending upon how much it has been. We shouldn't really talk about it just as damage, though, should we?
No,
because it's a thing that we hope to use to improve our general situation.
Give us more information on the
Right.
on the
Well, it's
whole
been real
story.
good to talk to you about this, Dick.
Pleasure to talk with you, Walter.
Our thanks to Dr. Caldecott for his very interesting and informative discussion on the application of radiation in the field of agriculture. From this sort of thing, we can develop the confidence that our research and development work will maintain the high standard of living that we now have in this country. Won't you join us in our next discussion on nuclear energy, Key to Tomorrow?
Nuclear Energy, Key to Tomorrow. Produced by WQED for the Educational Television and Radio Center, in cooperation with the Westinghouse Electric Corporation and the Atomic Energy Commission. The subject, agriculture. Your host, Dr. Warren F. Witzig, Manager, Reactor Physics. Special guest, Dr. Richard S. Caldecott, geneticist for the United States Department of Agriculture and the Department of Agronomy and Plant Genetics of the University of Minnesota. Technical advisor for the series, Mr. Edward R. Sanford. Engineering supervision by Frank Stuckman and Stephen Zorvis. Production and direction for the series by John W. Ziegler and Robert E. Potts. This is National Educational Television.